right. So this first question says a model of a process involving nucleic acids is shown in figure 1. There's figure 1. Which of the following best explains what process is represented here? So what do you see? I mean, this might be a little obvious based on the fact that we just recently talked about it, but what are you seeing here? No, not translation. It is DNA replication. How can you tell? Yes. Okay, so can you see if you can see what happens if you unmute yourself?
Hello.
Okay, say what you just said again.
Okay,
Mr.
because the way
you have
it's
to mute
shaped.
yourself.
What did you say, Selena?
Mr. Rudd, you're gonna have to mute yourself when
Oh,
she
I'm gonna
starts
have to mute
speaking.
myself if she talks. Okay,
Yeah.
I'll mute myself and then you talk.
Because of the way it's shaped. I'm sorry. And there's like a lagging strand. I just see that on the top too. You see it?
Yeah.
Did you mute your computer too, Mr. Rudd?
Yeah.
But yeah, there's also a lagging strand. But yeah.
Where? On the Google Meets? No, but I think I'm going to transition to Zoom. Oh, pros and cons. Okay, well, Lexi is pointing out the fact that you can see that there's a lagging strand on top because we can literally see that there are discontinuous arrows. So there's one arrow, and then after it, there's another one. And that's happening because on the lagging strand, we're kind of moving backwards. So we've got to kind of split up this process of replicating the DNA, split up the process of adding new nucleotides. That's why we call it the lagging strand. On the bottom, we can see it's just continuous addition of new nucleotides. So therefore, that is the leading strand. So before we even look at, and this is a multiple choice question, so before we've even looked at the answer choices, we already know what process is taking place here. So now we need to make sure that our answer matches up with what we have deduced already. So here are the answer choices. It says A, new DNA strands are being synthesized in the 3' to 5' direction from their DNA templates. B, new DNA strands are being synthesized in the 5' to 3' direction from their DNA templates. C, a new RNA strand is being synthesized in the 3' to 5' end from its DNA template. And D, 2 new RNA strands are being synthesized in both directions from their DNA template. D, as in. Okay, so Lexi is saying, I don't know if you can hear her, Selena, but Lexi is saying that she wants to rule out B as in boy because it says 5' to 3' direction. But she thinks that DNA replications happen in the 3' to 5' direction. What do you think about that?
I agree.
You agree?
Yeah.
And then now Lexi is saying she thinks it's A.
See, I was gonna say, like, is it A? I agree with her.
Well, okay, so why can we rule out C and D though? What's totally wrong about C and D?
because
Go ahead, Selena.
because RNA, it's not, I mean, it's in the process, but it's only like a primer. Like, they
Oh,
get rid
good
of
point.
it after
Good
a
point.
while.
Good point. So we know that RNA is a part of this process. I love that you mentioned that. It is a part of DNA replication, but it's only there as a primer. And also, I don't see any U's here. So in this image, we can't even see any primers. Yeah, there are only T's here. So RNA should not be a part of the answer. So good choice there. So you guys think it's A, but It's B. Look at the direction it's going. Look, literally look at the arrow.
But I thought that couldn't work. I mean,
We always, yesterday what I told you was that, we're adding, okay, so that's the lagging strand. Hold on a second. Okay, so the leading strand, 5 prime, and 3 prime is over here. Now keep in mind that the 2 strands always have to be anti-parallel. When I put a new strand here, this is its 5 prime end, and this is its 3 prime end. You have it written correctly. So we can only add on to the 3 prime end. Just keep adding on the 3 prime end. And if you look up here, you see an arrow is telling us we're adding on to the 3 prime end. And on both strands, we're adding to the 3 prime end. You see that? Selena, do you see the arrows, how the arrows point from 5 prime to 3 prime?
yeah, I see that.
Okay. So it doesn't matter if we're talking about DNA replication or if we're talking about transcription, we always add new nucleotides to the 3 prime end. Always, always, always. And that's because of the chemistry. We don't really need to get into it, but the 3 prime carbon is only available. The 5 prime carbon is not available to add new things. The 5 prime carbon is attached to the phosphate groups. The 3 prime carbon is free. So we can only add to the 3 prime. Okay, let's take a look at this one. Figure 1 shows some relevant details of a model of how a deoxynucleotide, in this case DTMP, is added to a growing strand of DNA. The features of this model provide evidence for which explanation of why all growing strands are synthesized in the 5 prime to 3 prime direction. So this is basically what I was just talking about. There's chemistry involved that prevents us from adding new nucleotides on the 5 prime carbon. Do you remember what I just said? Why can't we add things to the 5 prime carbon? Because it's attached to the phosphate group, and we can literally see that in these images. Selena, we just want to point out that it is it's a anti-parallel structure. So the 5 prime end of one strand is next to the 3 prime end of another strand. Sorry, I just kind of zoned out there. But let's take a look at the answer choices then. So A, the 2 strands need to be anti-parallel to bond properly. So keep in mind, let me just read the question again. The features of this model provide evidence for which explanation of why all growing strands are synthesized in a 5 prime to 3 prime direction. Basically, why are strands made in the 5 prime to 3 prime direction? A, the 2 strands need to be anti-parallel to bond properly. B, thymine and adenine would not bond properly if the strand grew from 3 prime to 5 prime. C, the translation of mRNA occurs in the 5 prime to 3 prime direction. Therefore, the growing DNA strand must also grow in the 5 prime to 3 prime direction. D, the phosphate group attached to the 5 prime carbon of the DTMP forms a covalent bond with the oxygen atom attached to the 3 prime carbon of the growing strand. What do we think? I've got an answer from Lexi now. Selena, what do you think? Since I'm sure you didn't hear Lexi.
Kind of stuck. I mean, seeing these sound good, but like.
Okay, so you want to limit us to C and D. Let's just take another look at those. C says The translation of mRNA occurs in the 5' to 3' direction. Therefore, the growing DNA strand must also grow in the 5' to 3' direction. Now, okay, so in response to that answer choice, I know I ruled it out the most recent time I read the question, but we do want to pay attention to those first 5 words of the question, which says the features of this model. So do we see anything in this model that refers to RNA?
No.
No, yeah. We're talking about a deoxynucleotide. So deoxy, that's DNA. And also we've got that key there that shows us T, C, A, and G. So no U's. So there's no evidence of RNA in this image. So if if this were a real test and you were just using your intuition and doing the process of elimination, then you would choose D. And that's also what Lexi chose. Right, Lexi? And that's the correct answer. Good. So again, we can only add new nucleotides in the 3' direction because the 5', I'm actually going to go over to the computer and circle it for you so you see what I'm talking about. But the 5' carbon is already attached to a phosphate group. Give me 1 second. So because of that, Selena, there is no opportunity to add anything else to the 5' carbon. We can only add to the 3' carbon. Okay, one more question. Figure 1 illustrates a model of the molecules involved in DNA replication and their placement relative to one another. Which of the following correctly explains where DNA replication will begin on the strand oriented 5' to 3', reading from left to right? So where is replication going to begin? Where are we going to start adding new nucleotides? So again, I don't want to look at the answer choices yet. Let's see if we can figure it out. It is referring to that general area, yeah. Well, I guess it's not numbered, but see how, let's see. Give me 1 second. Okay, so yeah, just try to describe it as best as you can in words, Lexi. Okay, yeah. Okay, this one we're gonna have to look at the answer choices to know. So A, DNA replication will be randomly initiated along the unwound portion of the DNA strand since base pairing will occur. B, DNA replication cannot occur since there is already RNA base pairing with the template strand. C, DNA replication will be initiated to the left of the RNA since DNA polymerase requires an RNA primer. D, DNA replication will be initiated at the site of the topoisomerase since that is where DNA begins to uncoil. What do you think, Selena?
Can I get like 1 more minute?
Yes. And in the meantime, I'm going to go on mute and Lexi, you can explain to me.
Okay, I think I got an answer.
Did you say something, Selena?
Yeah.
What did you say?
So I think I got an answer. I'm not sure if it's right though.
What do you think?
Hmm. I'm gonna say C, but I'm not sure if it's
Good.
right.
Yes, you are right. Good job. So we know, I think you mentioned it or Lexi mentioned it about the last one too. DNA replication needs an RNA primer. You were the one who mentioned it, I think. And Lexi was just explaining to me that, in the drawing that we made, the RNA primer is visible. And this is how DNA polymerase knows to come and actually start adding new nucleotides. So yeah, we need to start right to the left of that RNA primer. Okay. This is the last day you'll be in person, Lexi. What'd you say? So you're coming back? Uh, okay. So overview of protein synthesis. We're just going to quickly take a look at this. I don't need you to write anything down right here. Actually, there will be a couple things I'll ask you to write down. But we're going to talk about transcription and then translation. We'll talk much more about translation later on. So there are some differences between how a prokaryotic cell and a eukaryotic cell go through protein synthesis. There are some important differences. So here we're just looking at the overall pictures. Do you see any differences that we should point out?
Yeah, the nucleus. Well,
Good.
I think nucleus. Yeah.
Yeah, and that's what Lexi just said as well. It does not, the eukaryotic cell has a nucleus, the prokaryotic cell does not. What else do we notice?
Um, the eukaryotic cell looks a little bit more complex than the prokaryotic one.
Yes, so the eukaryotic cell is definitely more complex. It's got, go ahead. Yeah, so eukaryotic cells have what's called precursor mRNA, also known as pre-mRNA, whereas eukaryotic cells do not. It goes, and then also in a eukaryotic cell, we talked about this yesterday briefly. Um, the pre-mRNA has to be processed before it can become mature mRNA. Uh, in prokaryotic cells, it doesn't go through this processing. And we'll talk about the reason why, but you can kind of tell in this picture why that's happening. So good, those are the differences that I wanted to point out. Of course, they both start off with DNA. All cells have DNA. As you all correctly pointed out, the DNA is in the nucleus of the eukaryotic cell, but in a prokaryotic cell, which does not have a nucleus or any membrane-bound organelles for that matter, the DNA is just floating around in the cytoplasm. Now we want to talk about that pre-mRNA that Lexi pointed out. So only in eukaryotic cells do we find pre-mRNA. I do want you to write that down. And pre-mRNA is just mRNA that has not yet been processed. So this pre-mRNA is kind of like the very first product of transcription in a eukaryotic cell. Good? Okay. Then we're going to look at this processing as well. Oops, actually, before I show it, I don't think Lexi saw it. And I don't think you were here for this. You may have been. Oops, I skipped it, huh? Okay. I don't know if you were here for this or not, Selena, but what happens during processing? We can kind of see in the parentheses what's happening, but what happens when the RNA is processed? Yes. Good. Good. Degradation, yeah. Good. And then did you see anything about splicing? Did you write anything about that down? Okay. What did you write? Splicing. Good. All right, I'm going to explain all that to you now, Selena, but Lexi got it 100%. So mRNA processing involves 3 steps, 3 processes. Number 1, the cell is going to add what is called a GTP cap to the 5' end. So let me just quickly try to represent this.
What's that?
Can you see that? Yeah. So we've got an mRNA transcript here. It is going to be immature, so it's not yet been processed. Can you hear me?
You was going in and out. I can hear you
I
now.
hate this computer. So we've got an mRNA transcript here, and it has not yet been processed. So we would consider it an immature transcript. The first thing that the cell is going to do is add a 5' GTP cap. So what this means is that there's a 5' end of this transcript as well as a 3' end. Remember that those numbers help us to define the direction. So watch this. So we're going to add a 5' GTP cap for recognition purposes. We're going to talk about this later when we talk about translation. Then on the other end of the transcript, on the 3' end, we're going to add what's called a poly A tail. So it's literally like 200 straight A's, just adenine, 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 adenine. So this is called the
I can't see your screen anymore.
You can't see what?
I can't see you anymore.
The internet may have prevented us from seeing it. What should I do? Let's see. Let me know if it comes back. If you hover over my image and then you click on the pin button, it should say pin to screen. See if that brings it up.
Okay, yeah, I see you now.
So the poly A tail is just a lot of different A's.
Hold on, Mr. Red. I can't hear you.
I'm gonna let's see if this works. See if this works too. It's gonna connect me to your phone.
Mine?
I think you're the Galaxy, right?
Yeah.
Hold on. Hold on. It's okay. Pretty sure I got it to work a couple days ago. Let me see if it works for my Google Voice. Hello, Selena, can you hear me?
Yeah.
Can you hear me, Selena?
It
Okay,
was a lot of
cool.
feedback.
Let's see if this works. Hold on, I might need to leave my phone here.
No.
Are you talking?
He is.
Yeah, I can't hear you.
Hello.
Yeah, I don't hear him.
I can hear Lexi.
See?
There's no service down here. This is awful.
Exactly. Someone could die, Mr. Red. And then if 911 will never get the call.
Make this work. I really want to make this work because this problem comes up in my other classes as well. Let's see, show Bluetooth devices, add Bluetooth. Right now, no, you don't need it. Well, Selena, I'm just gonna have to stand in front of the computer every time I talk, so I apologize. Can you see what's behind me?
Yeah, just
Oh,
not
you're
behind
muted
your
now.
head,
I
but
muted
like,
you.
yeah.
Can you say that one more time?
I say, yeah, I can see, just not behind your head, but like I can see. Like, I mean, I can just take a picture of it.
The ones in the front don't work. The ones on the side do. Okay, so yes, Selena. We add this poly A tail. The poly A tail is there for protection because, as you can see in this image, the RNA has to travel from the nucleus through the cytoplasm until it can get to a ribosome. Doing that exposes it to different enzymes. And so when it gets exposed to enzymes, those enzymes are literally going to start chomping off at the end. We don't want them to chomp off the real message. We don't want them to actually start eating mRNA. So we're going to put all these extra A's on the end because we don't really care about those. If they get eaten off, if they start to degrade, it's not a big deal. But it's the real message, the actual mRNA transcript that we want to protect. Does that make sense?
Yeah.
Okay. And then the third process is called splicing. Splicing is a little more complicated, but essentially there are two different sections in the mRNA, in the immature mRNA. So let's just, oh, you can't even see that. Let's just imagine that there are red sections that are on this transcript. Those sections are sections that we want to keep, but as you can see, there are gaps in between them. The gaps are called introns. They're like the interspace. The actual red sections, the sections that we want to keep, are called exons. So through this process of splicing, we remove the introns. We don't want them to be there.
Okay, that makes sense. And then that you said that falls in with like the splicing stuff.
Yes, that is the process of splicing, removing the introns and then actually combining the exons. That is the process of splicing.
Okay, so it wouldn't be like removing the end of those tails, right? That's not that.
So by the time the 5' cap, the GTP cap, does not get removed. We need that for translation. We're going to talk about that a little bit later. The poly A tail is going to get bitten off as the RNA moves through the cell. So most of that will be removed by the time the transcript gets to the ribosome as well.
Okay, I understand.
Okay, cool. Did it change? Yeah. All right. So in prokaryotic cells and in eukaryotic cells, there has to be some travel taking place. You don't need to write this necessarily. You should, of course, remember, hopefully, from Unit 2 that we've got ribosomes that are located in the cytoplasm. We call those free ribosomes because they are just kind of floating around through the cytoplasm. They're not attached to anything. But we also have some ribosomes that are attached to the rough ER. Do you remember that? Yeah, they stud the rough ER. You can't see any ER in this. You can't see any endoplasmic reticuli in this picture, but we should just know that there are ribosomes located on the ER as well. And then we're going to go through this process of translation. Remember that translation does not necessarily form a protein. This is something that we'll talk about later as well. Translation forms a polypeptide. Translation does not form a full protein. The polypeptide is only the primary structure of a protein. We'll talk more about that a little later on. So one more thing, and I do want you to write this down. The reason why there is no mRNA processing taking place in prokaryotic cells is because the mRNA is immediately translated as soon as it is transcribed. So you can see it's being transcribed by RNA polymerase, and then it immediately goes into the ribosomes. So it doesn't need all this processing. It doesn't need to have a poly A tail added to protect it. It doesn't need the 5' cap for recognition purposes. It's just going to immediately start going through ribosomes to be translated. All right. So I'm going to ask you a question now. And this is a good question because I feel like it just gets you thinking in the way that the AP test asks you to think. So in this image, we can see that there are 4 ribosomes. Do you see that? Yeah. Okay. There are 4 ribosomes. They're lined up right next to one another. And the mRNA is moving through all 4 of them at the same time. Why do you think it's important that the cell has like a series of ribosomes located right next to each other, translating the same RNA? Why is that important? Okay, yeah, because you want to make sure you have some insurance in case there's a mistake. Why else though? So think about how can I provide a hint? No, okay, here's your hint. So the ribosome that's furthest to the left, that clearly like just started making the protein, it's making a section of the protein that the ribosome to its right has already made. And that second ribosome is making a section of the protein that the ribosome to its right has already made. What disappear? So I kind of think of it like an assembly line of sorts. If you're working on an assembly line, you're just putting materials together in the same way, one after another. There's going to be somebody to your right. You make something, and then it gets passed on. Similar things are happening here. These ribosomes are lined up next to each other so that this one strand of RNA can be translated multiple times at the same time. It's passing through all these ribosomes simultaneously, and this allows the cell to create a lot of protein at once. Does that make sense? It's making the same protein. They're all translating the same exact message. It's just happening faster because we've got so many ribosomes lined up. In reality, this is called a polyribosome, but the AP class doesn't call for that. So that's why I didn't put it in the notes. But I just think it's cool that the cell has this adaptation. Instead of just running it through one ribosome at a time, which would be slower, let's just line up all these different ribosomes, run that same RNA through all of them, and we're producing the same protein, but now we've produced it like 10 times using just one using just one mRNA. Okay. All right, so let's see. I'm going to ask you guys some questions here about the genetic code. Now, bless you. The genetic code is one of the best examples or best evidence that we have of the commonality of life or the continuity of life. It's one of the best evidences we have of evolution because almost all living things use the exact same genetic code. And so now you're wondering, what is the genetic code? So let's talk about it a little bit. So the genetic code basically tells us how certain bases, certain nitrogenous bases, A's, T's, C's, and G's, code for certain amino acids. So if each nitrogenous base were to code for one amino acid, how many amino acids can be coded for? If each nitrogenous base codes for one amino acid, how many amino acids can be coded for? Lexi just gave me the answer. Selena, what do you think?
Is it 4?
Yeah, it's 4. Because in that case, A codes for one protein, T codes for one protein, C codes for one protein, and G codes for one protein. That means that there are 4 total proteins possible. The math there is 4 to the power of 1, and that, of course, equals 4. Okay? What if 2 nitrogenous bases code for one amino acid? So we need a combination of 2 now. So instead of just A, I need AT. Instead of just T, I need TC. How many amino acids can come out of that? Good. Good. Yes, it is. Selena, do you have an answer?
No.
Okay. So Lexi said 16, and she's right. So if we need 2 bases for each amino acid, and of course, keep in mind, guys, that the order matters. So AT and TA are 2 different things because the order matters. So we could have AA, AT, AC, AG. Then we can have TA, TT, TC, TG, so on and so forth. So there are 16 total possibilities here. Now notice what the math is. 4 to the second power, 4 raised to a power of 2 equals 16. If we go back, we see that when there was only one base for one amino acid, then it was 4 to the power of 1. Now it's 4 to the power of 2 for 2 bases. So what's the rule? How do we determine how many amino acids are possible? Good, good. And I used N here, but you're right to have used X. So it's 4 to the N power, 4 to the nth power equals Y. And N would be the number of bases in the codon, whereas Y is the number of possible unique amino acids. Now, we're not saying the number of actual amino acids. We're just talking about the number of possible amino acids. But how long are actual codons? How many bases do actual codons have? Good. Actual codons have 3 nucleotides. They are 3 bases long. So that means how many amino acids are possible if you do that math using the rule we just came up with? 64. Oh, you're good at math, Lexi. I didn't realize you're this good at. So yeah, there would be 64. There should be 64 possible amino acids, but that's not how many there actually are. How many are there actually? Do you guys know? How many what? 3 more what? An amino acid? No, the codon is 3, like you said. So Every group of 3 codes for an amino acid. Every codon codes for it. No. Okay, this is kind of a random fact. There's no real reason you should know this. There are 20 amino acids in nature. How many? Yeah. Yeah. So there are 20 possible amino acids. But we just found out from Lexi's math that there should be 64 possible amino acids. So what is the discrepancy between 64 possible and 20 actual? Why are there 44 fewer than what we should expect? Any thoughts about that?
Is it because like I know what I want to say, but I don't know how to say it. Like,
let me show you this. Maybe this will help.
Oh yeah, I remember that. Like different orders.
Okay, you're on the right track. You are on the right track. If there were actually 64 amino acids, that would mean that every single combination coded for a different amino acid. That would mean that the order really, really matters. But what we see here is that not every combination gives you a different amino acid. For example, UUU, do you see UU in the very top left? UUU and UUC give you the same amino acid. UUA and UUG also give you the same amino acid. Why do you think that is? Why does that benefit the cell to have, to allow for certain changes? Say more about that. Do you have any thoughts about that, Selena? Lexi used the word flexibility, and that's a good word to use. Can you kind of pick up on that?
Nah, I don't really know how to explain it.
Can I erase this?
Yeah, you good.
And think about it like this. Before I actually do the science, let's think about like a real life example. So let's say you're at work, both of you have jobs. Let's say you're working the register. And let's say, for whatever reason, the power on the register is down. So you're having to do all the math mentally. Let's say that if you mess up by a single penny in your total count for the day, you get fired. That's it. There's no arguing. There's no flexibility there. That means that you have to be perfect in every single transaction. But if your manager tells you, if you're off by $5 or so, anything within a range of $5, you overcount by 5 or you undercount by 5. That's cool. We understand the power was down today, so you were doing all mental math. No big deal. You've got a lot more flexibility built in. The same thing applies to the cell here. For example, what I was just talking about. UUU is a codon that should give us the amino acid, I think it's called phenyl. I'm just going to use the abbreviation, PHE. But let's say something happens and the cell goes through a mutation. It gets mutated, which we're going to talk about later on next week. So let's say that last U gets messed up. Now we have UUC. The good thing about it is that if we have this mutation, the resulting amino acid is still the same. This is a good thing for the cell because it means that even if there are mistakes in this process of translation or in the process of transcription, the cell is going to still have a chance at producing the correct protein. If we change the amino acid with every little small change, that would be a bad thing. The cell would have to be perfect 100% of the time. In this case, we've got some built-in insurance. The cool thing about it is, let's say that there's a mutation and we get UUA. What amino acid do I get? I get LEU, yeah. So this is a different amino acid, but if we were to actually look at the chemical properties of these amino acids, they're different, but they're very similar. So again, the protein still has a chance at working. If we think about, remember, enzymes, enzymes have to be a very specific shape, and that shape is determined by their amino acids. So if we change even one amino acid, it could mess up the enzyme. But if the amino acid that gets changed is still pretty similar to what it should be, then the enzyme has a chance at working. Okay, so there are these built-in mechanisms that help to protect the cell. Insurance, basically. So these are. All right, cool. So now we get that. So one thing I do want you guys to do in your notes, though, I want you to define in your own words this phrase, synonymous codons. Synonymous codons. I'll leave this up and then I'll go back to the amino acid chart so you can kind of use that for your definition. Okay. So we're thinking about this phrase, synonymous codons. Good. That's a good definition for it. Codons that are different, but that still give you the same amino acid or similar amino acids. All right, we've got 15 minutes left. Let's see. Do I want to start this now? I don't think so, because this is.
I finished that thing, by the way.
Okay, cool. I do just want, let's just write this slide down, because this is kind of the overview of what we're going to talk about on Monday. Yeah. Well, look at the first bullet point. I think you know that already. Translation happens in ribosomes. You know where ribosomes are. I do want you to write down the second bullet point. Translation requires energy in the form of ATP and GTP. You have not written that third one down. There are 3 steps of translation. These are actually the same 3 steps for transcription Again, like I said, the AP curriculum doesn't really require us to go into detail with transcription, but it does require us to go into detail with translation. So the 3 steps are initiation first, then the second step is elongation, and then the third step is termination. Once you write that down, you are good. I'm going to stop the recording here. I did put those last 2 classes on YouTube, so hopefully you guys have a chance to check that out.